So, here I provide a brief ApiNATOMY update for January 2026, using as a driving example the renal connectivity model that we've just released for the Common Fund data ecosystem. I will start with a 3-minute recap on the evolution of the ApiNATOMY Toolkit. In the second half of this video, I'll walk through a model on flow routes along renal compartments that this toolkit has helped us organize. So, Part 1: Origins. The development of the ApiNATOMY Toolkit was initially driven by the need to create a fair knowledge scaffold over which to organize mathematical models of transport processes, and the multiscale route networks over which such processes occur. To meet such a need, we created representations of biological conduits, known as lyphs, that are assembled as layered cylindrical compartments. Each compartmental layer is linked to representations of biological materials that are annotated by standard reference ontologies to ensure fair semantic interoperability of the content they hold. Over the course of ApiNATOMY's evolution, we focused on adapting lyph assemblies to help represent 4 key constructs in physiology. 1. A specialized lyph as a primary functional tissue unit, or PFTU, that centers a short-range cylindrical diffusive field to a central long-range advective vessel. 2. Conduit arborizations as chains of lyphs representing trees, such as branched blood vessels, airways, or as in the case of the 3 chains shown here, chains of neuron segments embedded over a chain of spinal cord segments as part of the circuitry innervating the urinary chain. And for clarification, we note the layers in the spinal cord lyphs represent the cerebrospinal fluid in blue, the rexid lamina of the gray matter in grayscale, and the white matter funiculi in yellow. 3. Coalescent assemblies of primary functional tissue units that track the layered path of compartments for lateral exchange between long-range thoroughfares. In this example, the outer basement membrane layer of the epithelial and endothelial lyphs is shared. So here is a case of nested lyphs, where the ordered stack of tissue compartments acts as housing container to chains of renal and vascular cellular segments, which, in turn, act as housing container to chains of membrane channel segments. And 4. Assembling lyphs, their chains, and their coalescences into a coherent knowledge graph of multiscale flow routes to create the whole-body 2-map, an interactive homunculus of multiscale flow thoroughfares. So, let's put all this together by taking a deep dive into an ApiNATOMY model for kidney transport, utilizing apps from the ApiNATOMY Toolkit to navigate connectivity for endothelial and epithelial vessels in that organ. So, Part 2: A multiscale ApiNATOMY model for renal connectivity. In the ApiNATOMY Viewer app, we note 2 chains of lyphs extracted from 2 distinct components of the 2-map. The top chain spans blood vessels from the renal segmental artery to the renal interlobar vein, while the lower chain spans conduits from Bowman's capsule to the outflow of the urethra. Each rectangular flag represents the rotational unit of a lyph, and, as you can see now, holds compartmental layering information for each conduit. Double-clicking on any layer displays a knowledge graph that maps to OBO Foundry ontology terms, providing a granular semantic description of the material constituents for each compartmental layer. Of course, renal blood vessels and nephron epithelial tubing plot a tortuous path in support of the kidney's countercurrent mechanism. Therefore, keeping tabs on the lateral exchange routes between blood and the urinary space can be challenging. Our approach is to convert textbook knowledge into sets of conduits that engage in coalescent attachment, leading to lateral exchange. Hence the construction of the renal coalescent sets for the renal corpuscle, that is, the coalescence between glomerular capillaries and the visceral surface of Bowman's capsule, the vasa recta and the nephron segments of the loop of Henle, and the juxtaglomerular apparatus formed from the coalescence of the distal convoluted tubule and both efferent and afferent arterioles. For ease of visualization, double-clicking on a node representing coalescent sets opens an overlay showing every possible pair of distinct conduits engaged in a pairwise coalescent assembly. Each assembly provides the ordered sequence of compartments that have to be traversed for exchange between the luminal materials for that pair of conduit segments. To provide further compartmental detail for this lateral route in the renal corpuscle, chains of renal or vascular cell segments are straddled over their corresponding stack of housing tissue compartments. Specifically, each cell segment is embedded into a distinct tissue compartment and explicitly represents a distinct cellular domain in terms of both cytoplasm and plasma membrane. This explicit representation stems from the annotation of these cell segments with the appropriate gene ontology and cell type ontology terms to ensure that the polarity of each cell chain is appropriately and unambiguously declared. It is worth noting that this consistent creation of post-coordinated zip codes for nested cellular and tissue compartments sets the stage to automate the mapping of location-specific molecular network models across cells, their functional tissue units, and associated long-range thoroughfares. Prototyping the mapping of such networks relevant to drug pharmacokinetics and pharmacodynamics is currently underway. As preview, I am showing here a rather basic network for the pharmacokinetic handling of pravastatin by basolateral SLC and apical ABZ transporters in a renal epithelial cell. Further updates on this PKPD pathway mapping work, as well as releases of new tissue models in ApiNATOMY, will be the subject of our follow-up video releases. Thank you.